curious little champions, I welcome you all to Decode NCERT series, where today we are going to decode a chapter that carries 11 marks in your PU2 board examinations, which is none other than sexual reproduction in flowering plants, my dear children. And before starting off, again, something important to be said, that is, what is the purpose of this video? This video's purpose is to save your time. If you're someone who is revising or someone who is starting off with this chapter for the first time, I hope that you do not fall in the second category. But still, if you fall, so do not worry, you still have time. You're going to ace it. I trust you completely. Okay, so basically, here in this particular video, I will be telling you what is important, what type of questions have come, so that it becomes easy for you to relate and learn. I have just one request from you, that is, take out your NCERT, take out a pen, and if you have a sticky note, you can take that out as well. Okay, you don't need to highlight it with me. Okay, in this chapter, there are so many highlighted regions. I'll be telling you certain important questions which have come. You can write it down if you want. Okay, the questions only. Okay, and if you want the highlighted PDF, we will be making it available to you so that you don't need to highlight by yourself. If you're someone who reads through NCERT only, you don't want the PDF version, then you can think about highlighting later on. As of now, you do not need that. Just try to make your mind up here itself. Okay, just write the question, that is important. So let's get started. So sometimes it might get stuck because this chapter is really big. Okay. So from here the chapter starts, the introduction part, you can just give it a read, a quick read, okay? You don't need to invest a lot of time into it. From here starts what is important. First thing, the male reproductive system, my dear children, very much important. Okay, so it is known as the stamen. Stamen has two parts, anther and filament, just as mentioned over here. And then they have mentioned that the anther is bilobed. It has two lobes. One lobe is this, another lobe is this. Then they have said that it is dithecous. So it has one theca, one theca, one theca, one theca. It is dithecous on both the sides. Okay. Now over here, in each theca, you will be finding what? Microsporangia. This microsporangia are going to form into what? Pollen sacs. So this is where the pollen grains are going to be produced. In each theca, you have microsporangia. Now they have mentioned about the four walls of microsporangia, which is very much important. Okay, epidermis, endothecium, tapetum, spore, and everything. Okay, middle layer as well. So they will ask you this question also. That is, the cells of the tapetum, it possesses dense cytoplasm and it has more than one nucleus. This is a unique feature. All of the cells of our body, or basically plant's body, has one nucleus. No. So how can something has two nucleus and also has dense cytoplasm? What is the use of this? So tapetum has dense cytoplasm for nourishing the growing pollen grains. Where are the pollen grains growing? Inside the microsporangia, there is an area that is known as sporogenous tissue. Here you have so many cells which are going to undergo meiotic division, which is going to form microspore tetrads, as in 4 microspores. These microspores are later going to form pollen grains. So this whole thing, if you do not know the flow, you are not going to understand. They will write you what are the events, they'll give the question, that is, what are the events of microsporogenesis? Write it down. Write it down in the sticky note. What are the events of microsporogenesis? Very important question. Okay, you have to tell about all this thing. Over here, this picture, that is transverse section of young anther, very much important. Transverse section of young anther. This here you have to mention the layers and everything. Only if you have understanding, you would be able to label it. That is why I told you, if you have any doubt regarding the concepts, you can go watch the Brahmastra series. It will really help you so well. Okay, now going ahead. I told you what happens to the microspore tetrad. They are going to dehydrate, and they are going to form individual pollen grains. Now, one very important thing, what is the male gametophyte? It is the pollen grains which are the male gametophyte. What do you mean by male gametophyte? The ones that will be forming the male gametes. So microsporogenesis is happening. Microsporangia is not the male gametophyte. No, it is the pollen grains. So after microsporogenesis is done, you get pollen grains. The pollen grains has the male gamete, my dear children. So on the basis of this, questions can come like this. Select the correct sequence of events in microsporogenesis. Okay, so you should be knowing there is sporogenous tissue. In that you have microspore mother cells. All the cells, as I said, they have the ability to undergo meiosis. So cells are known as microspore mother cells because they are going to form microspore. So later you get microspore tetrad. As in 4 of them, they are going to move away. You get microspores which are known as the pollen grains. Very much important. So this is a question. Okay. Now going ahead. Obvious. You know what? I am recording this video for the 3rd time now and it's getting stuck there. I don't understand why. But let's just wait. Okay. And also violet loads. Violet loads, let me tell you about this thing. It's very much important. That is, if you want to understand the structure of microsporangia, you can look for this particular image. It has more clarity than this. Okay, so epidermis is mentioned, then endothecium is mentioned, then you have middle layer, then you have tapetum, okay, and then inside the tapetum you have the microspore mother cells which are mentioned over here, just like this. Okay, so finally it is loaded over here. Again, now we are talking about the structure of the pollen. So you should be knowing that it is having a outer layer which is known as exine, then there is an inner layer which is known as intine, and there is an area where there is no exine intine. There's a space which is not no exine, which is known as what? Germ pore. What is the use of germ pore, my dear children? Through here only the pollen tube will be moving out, carrying the male gametes. Okay, so over here what is the exine made up of? Very important question. Over here you see sporopollenin. This is a thing that from which exine is made up of, and this is a very tough component which can withstand what? High temperatures and strong acids and alkali and everything. So it's the most resistant organic matter. It's a very important question that I am telling you over here. Okay, now the inner wall, as I said, it is intine. Apart from that, you're talking about the walls, but what is in there? In the cell. So cell basically is divided into two parts. There is one vegetative cell and there is one generative cell, my dear children. Okay, there is one vegetative and there is one generative cell. Now listen to me carefully. The vegetative cell, it is there. Okay, it helps in the formation of pollen tube and everything. Generative cell which is there, it can be still it has not formed the gametes. The generative cells, it can undergo division to form two male gametes. It can form two male gametes. Now this process can happen later on, that is when the pollen grains comes and lands on the stigma, or it can happen before only. Okay, when the pollen grain is released at this point of time itself, the generative cell can be divided into two male gametes as well. So here they have mentioned something, that is, in over 60% of the angiosperms, pollen grains are shed at this two cell stage. That means vegetative cell and generative cell. There is no division happening. Okay. Later, in the remaining species, that is 40%. Okay, remember the percentage, 60 and 40. In the 40%, generative cell divides mitotically to give rise to two male gametes. So it will be released in three cell stage, one vegetative cell and two male gametes. Very, very important. Now what type of questions are coming? Over here I have mentioned the question. The most resistant organic material present in the exine of the pollen grain, sporopollenin. Sporopollenin is one of the most resistant materials known. And where is it found, children? It is found in the exine. Either way, the questions can be coming. Now, talking about the pollen allergens, things like that, and things like what is a contaminant, okay, which is being there in our country. So, Parthenium or carrot grass that came into India is a contaminant which came with what? Imported wheat. So, Parthenium, this name is important. Okay, and the question can come on the cells of the male gametophyte. What is male gametophyte? Pollen grain. Okay, what are the cells? Vegetative cell and generative cell. What is the shape of the nucleus of the cell? So over here it is kind of round, whereas here it is kind of ovular in shape. Okay, so either way, these questions can also come. Such questions have come. Okay, such questions have already come, which is why I am explaining it to you. Now, going ahead, what do we have? Yes, pollen consumption has been claimed to increase the performance of athletes and racehorses. This is also important. Somehow I did not highlight, so I'll highlight it right away. Ayyo, why is it not highlighting? Just a second. Highlight, macha, highlight. Okay. Okay, anyways, from here to here. Okay, it is important. Now. Again, in some cereals such as rice and wheat, pollen grains lose viability. Here you are talking about the viability. So in some, the viability lasts for longer time. Okay, it will be able to fertilize, it will be able to do everything for a longer time. Some viability is really less. So in some cereals such as what? Rice and wheat, the pollen grains lose viability within 30 minutes of their release. So it is only viable up to 30 minutes, after that is gone. Okay, and in some members of Rosaceae, Leguminosae and Solanaceae, they maintain viability for months. So these examples are really important. Okay, and how to store pollen grains? It is stored for years in liquid nitrogen, which has minus 196 degrees Celsius. Okay, that is important piece of information. Now we talk about the female reproductive system. Everything over here is important. You have to read it, which is why I mentioned like this: monocarpillary, multicarpillary, this carpus, that carpus, everything. So questions can be coming like this also. Define the following terms: staminate flowers. What do you mean by staminate flowers? The one that has just the stamens. That means you're talking about the male flowers. Pistillate flowers, that has, pistil is what? The female reproductive part. So it is the female flower. It has stigma, style and ovary. everything. So each and every information over here is important. You would have to make the diagram as well. They would ask you for the diagram, not this diagram, but generally a diagram of the ovary and everything. Okay, where is the micropyle? You should be knowing that. Okay, where is the micropyle? This is where, from where the pollen tube will come enter. Okay, so all this information is really important. So explain the structure of the megasporangium. What is the megasporangium? Basically talking about the ovule. Okay, you are talking about the ovule. So you should be knowing about the structure of the ovule, which is what I mentioned over here. So ovule is surrounded by this thing, outer integument, inner integument. Then you have nucellus, okay, and everything. There is micropylar end, there is chalazial end, funicle is there, there is hilum. Everything is very much important. Okay, what is the female gametophyte, my dear children? Embryo sac is the female gametophyte. It is made inside the ovule. Okay. Now they can ask you the structure of this thing, that is an embryo sac. Very important diagram, this diagram. So as I said, everything over here is important. You cannot skip anything until this particular stage. Okay, you have to read, they will ask you about the events of megasporogenesis. Okay, megaspores or megasporogenesis. Okay. Now over here, pollination, you should know what is pollination, and kinds of pollination. You have three different: autogamy, gytenogamy and syngamy. Xenogamy. Okay, so autogamy, what is their transfer of pollen grains from anther to the stigma of the same flower, that is autogamy. Now autogamy in such flowers requires what? What are the requirements for autogamy? You should know that. Synchrony in pollen release and stigma receptivity. Also the anther and the stigma should be what? Should lie close to each other, everything. So some plants like Viola, Oxalis and Commelina produce two type of flowers. What are those two type of flowers? Chasmogamous flowers and clistogamous. As in chasmogamous as in open flowers, clistogamous as in closed flowers. So in a flower, in the stem only, in the lower part they have closed flowers, in the upper part they have open flowers. What is the use of closed flowers? Very much important. What is there in closed flowers, my dear children? If it is closed and if it has both the male and the female parts, they are going to self-fertilize, they are going to self-pollinate, and anyhow they will be getting seeds. Fertilization will happen, seed formation will be taking place. So we say in clistogamous flowers, you produce assured seed set even in the absence of pollinators, even if pollinators are not there. Okay, because it's closed. Why there's a need for pollinators? You will get assured seed set. What type of flowers? Clistogamous. What are the examples? Which plants have this? Viola, Oxalis, Commelina, etc. Okay. Now over here, gytenogamy, again very much important. They will ask you for the difference between gytenogamy and xerogamy. Okay. So name the type of pollination that brings genetically different types of pollen to the stigma. Genetically different. Okay, that means you're talking about xenogamy. In xenogamy, you have two different plants. In two different plants, if pollination is happening, obviously they are different genetically. Talking about gytenogamy, gytenogamy means two different flowers of the same plant. Two different flowers of the same plant, and that means genetically it is same. Genetically it is same. Autogamy is basically when you're talking about the same flower. Okay, you're talking about the same flower. Gytenogamy as in two flowers of the same plant. Xenogamy as in two plants itself. Okay. Now differentiate between gytenogamy and xerogamy, so read through it, very much important. Agents of pollination, majority of the plants use biotic agents. So biotic agents as in living agents are more useful, okay, than abiotic ones. Now in abiotic, which one is useful? Abiotic, pollination by wind is more common amongst the abiotic pollinations. Okay, they can ask you, write any 4 characteristics of flowers pollinated by wind. So you should be knowing about all this thing. Pollen grains are light and non-sticky so that they can be transported in wind currents. They often possess well exposed stamens so that they can capture the pollen grains. Large and often feathery stigma, again to capture. Okay, and then what? Wind pollinated flowers often have a single ovule in each ovary. They do not have multiple ovules. Why to waste energy? Anyways, we do not know pollination will happen or not. Okay, so you cannot put energy into making multiple ovules. That will be wastage of energy. So they usually have one ovule only. So the question can come, what is that type of pollination where the flowers have only one ovule in the ovary? So it is wind pollinated flowers. Okay. Now, so again, tassels in the corn cob that's seen over here represents what? Tassels that you see over here represents what? The stigma and the style which wave in the wind. This is very much important. So wind pollination is quite common in grasses, again very important piece of information. In grasses also you see wind pollination, my dear children. Okay. Now talking about water pollination. So mode of transport for the male gametes among the lower plant groups such as algae, bryophytes and pteridophytes. So they have the water pollination thing. Some bryophytes and it is believed that some bryophytes and pteridophytes, okay, that is already mentioned. Now over here, examples, Vallisneria, Hydrilla, Zoostera, everything is given over here. Some basically release on the top of the water, some in the water and things like that. You should be knowing who all are there. So in a majority of aquatic plants such as water hyacinth and water lily, the flowers emerge above the level of water, even though they are in water. Even though they are in water, they do not prefer water pollination. They are above the level of water, right? So they prefer wind pollination and stuff like that, or insect pollination. So basically are pollinated by insect or wind as in the land plants. But when you talk about, so when you say water hyacinth, water lily, you expect water pollination, but they are showing what, wind pollination or insect pollination. But Vallisneria, the female flowers reach the surface by the long stalk, and the male flowers or the pollen grains are released on the surface of the water. They carry it passively in Vallisneria. In the next plant, it will be different. In another group of plants, that is sea grasses or basically Zoostera, the female flowers remain submerged in water, as in inside the water only. The pollen grains are released in the water. Okay, and the pollen grains in such species are long, ribbon-like, so that they can move easily in the water itself. Okay, so that is different. They are submerged and they release inside only. In most of the water pollinated species, pollen grains are protected from wetting by mucilaginous covering, so that it does not get damaged, dissipated and things like that. Okay. Now talking about insect pollinators, they would ask you the type of insects or type of animals also. So you know what the insects, honeybee and things like that. Animals you do not know. So animals, even large animals such as what, some primates like lemurs or arboreal rodents or even reptiles, lizard and garden lizard, they all act as biotic agents. Again a question can come, that is basically, okay, now over here the question is asked, what are the features of insect pollinated flowers? So you should be knowing this. This is a very important question that came. Picture related to pollination is given below. This was the picture that was given, okay, where an insect is sitting on the flower. Will this pollination confirm fertilization? You should be knowing about that. So you cannot say that pollination always confirm fertilization. If the pollen grains form the pollen tube, that's when fertilization and everything will happen. So obviously no. What are the floral rewards provided by the plants to the insects to revisit? So you should be knowing that the floral rewards are what, basically nectar and pollen grains and things like that. Okay, mention the characteristics of a flower in this pollination other than the floral rewards. So you should be knowing that the flowers are large, colorful, fragrant and rich in nectar. Okay, when the flowers are small, they are arranged in a conspicuous manner which makes them look brighter, bigger and things like that, so that the insects can see and come over here. Okay, the flowers pollinated by flies and beetles secrete foul odors because that's what attracts them. To sustain animal visits, what do they do? Nectar and pollen grains. Very important thing. Now apart from this, in some species, floral rewards are in providing safe places to lay eggs as well. That is also a reward, my dear children, to give a safe space for laying their eggs. What is the example? That is, example is Amorphophallus. That is the tallest flower. Basically it is 6 feet in height. Okay, a similar relationship exists between species of moth and the plant yucca, where both the species moth and the yucca cannot complete their life cycle without each other. So moth wants to lay its egg over there and yucca is dependent on it for pollination to happen. Okay, so this is very important thing. Obligatorily, call this as obligatory relationship. They don't have an option there. So in turn gets pollinated by moth. The larvae of the moth comes out of the egg as the seed starts developing. So yucca and every information, Amorphophallus and the moth that is there, you have to remember it. Okay, now explain the any three outbreeding devices. So you should be knowing what do you mean by outbreeding, what does it cause. So if not outbreeding, why do you need outbreeding? That is the thing. So if self-pollination is happening again and again and again and again, inbreeding depression happens. The plants would not be able to produce fertile offsprings and stuff like that, which is why the plants itself have made some outbreeding devices. That is something that could make the plants pollinate between different plants. Okay, there is no self-pollination happening. So that is what you mean by outbreeding devices. You should be knowing what are those. So in some species, pollen release and stigma receptivity are not synchronized. So if it is not synchronized, you don't expect pollination to happen. Next thing, in some other species, the anther and the stigma are placed at different positions. That is there. And now the third device is to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollen from fertilizing the ovule. So this is a genetic mechanism which does not allow, okay, it does not allow pollen tube to form. So these are the three. Apart from that, production of unisexual flowers. If it is unisexual, obviously cross pollination has to happen. If stamen is being there, the stamen is there, it cannot just release pollen and sit there, right? The pollen has to go land on somewhere else. So it is basically going to help promote cross pollination as well. Okay, so you should be knowing this very important thing. Examples, that is castor and maize. They are monoecious. It prevents autogamy but not gytenogamy. Okay, they are monoecious. It prevents autogamy. That means basically in the same flower, you would not be having this thing, both the sexes. You will be having different sexes. Okay, in one flower you have one sex, in another flower you have another sex, but in the same plant. So in the same plant, if between different flowers it is happening, it is known as gytogamy. What do you mean by autogamy? If in the same plant or same flower, in the same flower, if pollen grains are moving here and there, that is what you mean by autogamy. So example is castor and maize, they are monoecious, but they prevent autogamy but not gytogamy. Important piece of information. Okay, now pollen-pistil interaction, what is important over here? If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma of the pollen growth in the style. So you should be knowing this. What kind of dialogue is there? Chemical dialogue is there in between them, that is pollen and the stigma and everything, so that compatible, if it is compatible, that's when the pollen tube formation will happen, otherwise it would not happen. So you should be knowing about that as well. Now pollen tube carries the two male gametes from the beginning. Pollen tubes, after reaching the ovary, enters the ovule through what? Micropyle end, remember that, and enters the synergids through the filiform apparatus. So first thing that it enters is synergids. Okay, so in the embryo sac there are synergids, there are antipodal cells and everything. I will be talking about that as well, don't worry. Now all these events from the pollen deposition on the stigma until the pollen tube enters the ovule, what do you call this as? Pollen-pistil interaction, my dear children. Now, artificial hybridization is there, very much important. They will ask you the steps for artificial hybridization, what do you mean by that? So it is one of the major approaches of the crop improvement program. Okay, you don't want cell pollination to happen. You want a type of plant that you are looking forward for. So what do you do? You collect the pollen grains and you put it on the stigma, and then basically you wait for the progenies to be made. So over here, if at all the plant already has anther and everything, you have to remove the anther so that it does not undergo self-pollination. You call this as what? Emasculation. And then you basically cover it up so that no other pollen grains can come on landed, that is bagging. So between that you put the pollen grains and you bag it completely. So these are the steps, you should be knowing it. Double fertilization, again one very important thing. All the events over here are important. Okay, that is basically there are two male gametes, right? One male gamete comes and fuses with the egg cell, it is known as syngamy. The process is known as syngamy, and what does it produce? Zygote, my dear children. Okay, another male gamete is there, it will go and fuse with the two polar nuclei, and when it fuses with the two polar nuclei, what do you get? The primary endosperm nucleus, my dear children. Okay, so 2 times fertilization happening, which is why it is known as double fertilization. Okay, and here in primary endosperm nucleus, there are 3 fusions happening. The nucleus, there are 3. 2 polar nuclei, that is 2 nucleus, and one nucleus from the male gamete. So 3 nucleus, which is known as triple fusion. This is known as syngamy, this is known as triple fusion. Both are done together is known as double fertilization. The central cell after the triple fusion becomes primary endosperm cell and develops into endosperm, very much important. So picture of a mature angiosperm embryo sac is given below, and answer the following. They will give you the picture and they will ask you the questions as well, or they can ask you to form the embryo sac. Which cells, nuclei of the embryo sac produce zygote and the primary endosperm nucleus? You should know that. So what produces zygote in the embryo sac? It is the egg cell. The female gamete fuses with the male gamete to form the zygote. What forms the primary endosperm nucleus, my dear children? The polar nuclei. The 2 polar nuclei comes in contact with the male gamete again to form the endosperm nucleus. Okay. Now, what is the ploidy of antipodal cells and primary endosperm nucleus? So antipodal cells, what are those? Is it mentioned over here? No. It is mentioned in the embryo sac. So all this ploidy as in the content of DNA. Okay, content of DNA. Always remember, in the embryo sac, they always have haploid content. Okay, embryo sac as in where the female gamete is there. The female gamete has to have half the number of chromosome. The male gamete also will have half the number of chromosome. Together they come and make double the number of chromosome which was there initially in the parent, right? So everything that is there in the embryo sac will have half the number of chromosome. So antipodal cells, synergids, egg cell, everything have half the number of chromosome, that is haploid, that is the ploidy. Haploid is the ploidy. Primary endosperm nucleus is formed when there is 2 nuclei and there is 1 more nuclei. So this nuclei had half, this nuclei had half, okay, that is n number of chromosome, over here. This is also going to contain n, this is male gamete, this is also going to contain n number of chromosome, n. This is known as triploid, okay, 3n, this is known as triploid endosperm, primary endosperm nucleus, okay. So why endosperm development precedes embryo development? Why? Endosperm development precedes embryo development. Why? Because the endosperm will be providing nourishment for the zygote to form the embryo, okay. So know about that. This image very much important. What is this image? This is dicot, this is a dicot embryo and this is a monocot embryo. So dicot embryo and LS of a monocot embryo. So they can ask you LS of embryo of grass. So grass is a monocot. Monocot as in they have just one cotyledon. Dicot as in they have two cotyledons. So over here you see these are the two cotyledons that they are having. Dicot. This is the one cotyledon that this guy is having. This is known as monocot. So grass is a monocot. It's an example of a monocot. So remember like that. Okay, do not be worried. Now, endosperm may be either be completely consumed by developing embryos such as in pea, groundnut and beans, very much important, and in some, it may persist in the mature seeds such as castor and coconut. So, I will tell you one thing, in dicots, it is completely consumed because they are using endosperm to form the two cotyledons. Okay, so it is completely consumed. So everywhere where you can break them into two, for example pea and everything which you can break into two, that means they are dicot and they will be completely utilizing their endosperm for formation of cotyledons. Whereas when you talk about monocots, okay, they cannot utilize completely, they do not utilize it completely, they have some endosperm still persisting, they just make one cotyledon, okay, which is what is known as monocot. So castor and coconut, they are monocots. Groundnut, pea and beans, everything which you can break into two is dicot. Remember like that. Now, embryo develops at the micropolar end of the embryo sac, very much important. The correct sequence of embryonic development in angiosperm. So, very much important again. So, over here you have a picture. Okay, you have zygote, globular embryo, heart-shaped embryo, mature embryo and things like that. You should be knowing the flow over here. If you are not knowing the flow, you are going to mess it up big time. Okay, so here also they have mentioned. You have pro-embryo and then you have globular embryo, heart-shaped embryo and then mature embryo. So, go know the flow, the questions have come. Okay. Now what? This all thing is important. Okay, you are talking about the dicot embryo completely, you are talking about the monocot embryo. Draw an LS of the grass embryo, which is this, and you have to mention all the parts, that is scutellum, coleoptile, shoot apex, this apex, that apex, everything. So if you are not understanding the concepts over here, you won't be able to draw it there. That is why I said watch Pro Master series, it will be important for you. Children, I hope that you do not mind me being really fast over here, because I don't want to waste your time. This is for revision, right? So let's wait. Yes, so we go ahead. So all this structure, as I said, monocot, dicot, embryo, you should be knowing the structure. Okay. Now the next thing, you're talking about seed. So there are two types of seed, albuminous seeds and non-albuminous seeds, which are just hidden after behind this. Okay. So albuminous is where you have still some amount of endosperm still left. That means you're talking about monocots. Okay, non-albuminous means they do not have anything left behind. That means you're talking about dicots. Okay, one very important thing, examples they have given you once more. Apart from castor and coconut, they have given you more example, that is wheat, maize, barley and everything. Remember that. Now one very important thing over here, where is it? Yeah, so in some seeds such as black pepper and beet, remnants of the nucellus are also persistent. So nucellus is a part of the female reproductive system, okay, in the ovule itself. So usually the nucellus is completely utilized, but over there the nucleus is persisting. Okay, and this nucellus is also providing nutrition. So this persistent nucleus is known as perisperm. So question usually comes on perisperm as well. And what is the ploidy of perisperm? Since it is a normal cell that is found in the ovule, you say it is diploid. You say it is what? Children, diploid in nature. Okay, this cells of the nucleus, one cell of the nucleus will undergo meiotic division to form haploid embryo sac and stuff like that. Okay, but as of now, the nucleus, when you talk about that, it is diploid. So obviously the perisperm which is there, it will also be diploid. Now differentiate between pericarp and perisperm. Pericarp is the outer covering of this seed. So whenever fertilization happens, we say the ovule, the fertilized ovule is going to form the seed, okay, and the fertilized ovary is going to form the fruit. So outer you have fruit and inside you have what? The seed. So the covering of the seed is known as pericarp. It is known as what? Pericarp. So pericarp and perisperm is different. For perisperm, there are lots of questions that have come, that is endosperm persists in mature seeds in what plant? Black pepper and beet. Two examples only. Remember that. Ploidy of perisperm present, I told you diploid. Differentiate between pericarp and perisperm, another question. So many questions come. Residual persistent nucellus present in some seeds is called, again, perisperm. Now, the micropyle remains as a small pore. This is very important piece of information. Okay, the micropyle is a small pore that you see on the seed coat. In the seed coat, you see a small pore, right? That is the micropyle. This facilitates the entry of oxygen and water into the seed during germination. Okay. Now, apart from that, as I said, pericarp and everything. So, in some, the fruits may be fleshy as in guava, orange, mango, or it may be dry as in groundnut and mustard. Okay, that is there. Now, false fruits and true fruits, you should be knowing. False fruits as in from ovary, apart from ovary, from somewhere else, if fruit is being formed, you call them false fruits. For example, you have strawberry, cashew and apple and everything. Okay, from where it is forming, so thalamus also contributes to the fruit formation, which are known as, okay, anyways. True fruits are the ones that is developing from the ovary, remember that. Then there is a parthenocarpic fruits is also there. Okay, this picture is also important. They can ask you to label it. Okay, they can ask you to label it, so please know. Okay, this is a monocot, scutellum is mentioned, that means it's a monocot. Okay, so you see in the strawberry, thalamus is one thing that forms. Here also is a thalamus that is going to form the fruit. Okay, here also thalamus, strawberry, here in apple also it's a thalamus. Thalamus is a supporting structure which you have. Beneath the flower you have from bigger area, okay, one swollen area, you call this as thalamus. Cool? Now, parthenocarpic fruits, again, such fruits where fruits are being found without fertilization happening. This is important. Do not confuse this with apomixis. Apomixis is when seed is being found without fertilization. Here the fruit, the ovary, it is undergoing certain changes, that it is formed fruit without fertilization. Okay, it happens in what? In banana and things like that, and we can actually induce parthenocarpy as well. We can make the plants do parthenocarpy if we give certain hormones such as auxin and stuff like that. So they will become a fruit. Okay, the ovary will transform into a fruit without fertilization. Okay, so here, since fertilization is not happening, do you see seeds? No, you would not be finding seeds, my dear children. In parthenocarpic fruits, you do not find seeds. So that is one very important thing. So what is parthenocarpy? Give an example, you should be knowing. Parthenocarpy is what? Where fruits are developing without fertilization. Give an example, it's about banana and everything. You can induce parthenocarpy, everything is important. Now, over here, seeds, about seeds, lot of things are given, and one very important thing that is given over here, okay. Seeds offer several advantages to angiosperms. What are the advantages? First and second advantage is given over here. Just go through it once, okay? I won't say invest too much of your time, but just go through it once. Now, this is important, comes in MCQs, okay? So, there are several records of very old yet viable seeds. So, oldest is that of a lupine, that is Lupinus articus, excavated from Arctic tundra. You should be knowing that. This is the oldest, okay. Now, the seed germinated and the flowered after an estimated record of 10,000 years of dormancy. That means it is viable for that long, okay. The recent recorded 2,000 years old viable seed is that of date palm, that is Phoenix dactylifera, okay, discovered during the archaeological excavation at King Herod's. So, they can ask you what is that seed that was viable up to 2,000 years. So, it is date palm, Phoenix dactylifera. Remember the names: date palm, Phoenix dactylifera, lupine, that is Lupinus articus, it is the oldest, okay. It stayed for 10,000, this stayed for 2,000. Usually question comes from Phoenix dactylifera, but you can expect from that also. Now, going ahead with the last topic of this particular chapter, that is Apomixis, very important topic. Every time a short question has come, you cannot miss this particular topic, okay, and this is a confusing topic. So, if you want to understand the concept, go watch the video, okay. In the NEET videos also I have explained it very well. If you're someone who's finding difficulty in understanding Brahmastra series, okay, or you're scared of seeing the questions, go watch the NEET series as well, but it is time taking, which is why I'll recommend you to watch the Brahmastra itself. I will make you understand that, understand all your, take away all your confusions in that particular series, okay. So, watch it. Now, so apomixis is where, as I said, seed is produced without fertilization. So, you might say, how seed is produced without fertilization? Seed will only form if fertilization has happened, right? But in apomixis, things are different. Okay, so apomixis is seen, and also citrus, mango, everything is an example of polyembryony, apomixis, everything is important over here, my dear children, and they can ask you what is the use of apomixis seeds. Okay, so you can say basically in apomixis, in apomixis there is no fertilization, no? And Apomixis is like asexual mode of reproduction. That is also one question that comes. Okay, Apomixis has high similarity with asexual mode of reproduction. So Apomixis is a form of asexual reproduction that mimics sexual reproduction. So it is mimicking sexual reproduction. Why? Because seed formation is taking place. In asexual reproduction, seed formation does not take place. But it is actually asexual reproduction only. Why? Because there is no fertilization happening. Okay, and if no fertilization is happening, for example, I have a plant and it has all the features. So I want to create clones of this, and I would prefer asexual reproduction to prepare clones of it. If it is sexual, then there will be gametic changes, genetic changes, so it will be a huge lot of trouble. So Apomixis would give me what basically the desirable traits that I want. So I can, for example, I have a hybrid plant. Now, if I let this hybrid plant go pollinate with other plants, it will create new things. I don't want that. But if I create Apomixis seeds of this particular plant, then I just have to, I'm just making clones of the same plant. So it is highly useful for the farmers and everyone. So with this, we are completing the chapter. Okay, and this is what is important. You can invest your time and energy into studying this. I know you might have felt that it has went so long, or basically all the parts of this chapter is important, but at least you now know what is more important. Everything is important for sure in this chapter, but what is more important is what I said. Okay, and I've given you a question, go through it carefully. Okay, and I discussed this answers for the question in Brahmastra series. You can watch that as well. And with this, we are ending today's session. I hope it was helpful for you. I tried my level best to highlight and put the questions and everything in a correct manner so that you don't miss even a single piece of information and you don't miss a single mark over there. The other day I saw a message, or basically a comment, saying that that the person got really good marks in biology, and I was really happy seeing that. Everything matters, each and everything matters to me a lot. So thank you for all your love, my dear children. Don't forget to subscribe to our channel, that is Diksha Karnataka. Bye bye, take care.